Hello, I'm Essay, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
The 1965 work by Elizabeth Bugie and Selman Waksman, titled Isolation of New Antibiotic Substances, represents a fascinating document from a mature phase of the antibiotic revolution, arriving 2 decades after their foundational collaborations that yielded agents like streptomycin. This publication offers a lens into a scientific program at its zenith. Grappling with the very success it had created, it is a work of immense methodological confidence, a codification of a discovery paradigm, and a subtle acknowledgement of the emerging challenges posed by microbial resistance. This essay will provide a direct, section-by-section evaluation of the internal claims, arguments, and evidence presented within this specific academic contribution, assessing the work on its own terms as a product of its distinct intellectual moment. The analysis will proceed systematically through the paper's structure, examining the logical and evidentiary foundations of its propositions and elucidating the conceptual framework that underpins its entire investigative enterprise. Section 1, the intellectual framing of a continuing quest. The paper's introductory section articulates a rationale born from experience. It posits that the continued exploration of soil microorganisms for novel antimicrobial agents remains a primary imperative for therapeutic advancement. The central claim here is twofold: that the vast microbial diversity of the soil is a reservoir of untapped chemical novelty, and that the established Waksmanian screening methodology is the most efficacious engine for its discovery. The argument is presented with the authority of established success, referencing the lineage of antibiotics derived from actinomycetes. The authors build their case on the premise that nature's chemical arsenal, developed through eons of microbial competition, far exceed the imaginative capacity of synthetic chemistry as it existed in the mid-1960s. The evaluation of this claim reveals its deep roots in an ecological perspective of microbiology. The argument's strength lies in its empirical foundation. The historical record of the previous 20 years provided ample evidence for the productivity of this approach. The work frames the problem of antibiotic discovery as one of systematic, persistent exploration, akin to a form of natural history married to biochemistry. The authors contend that the challenge is one of scale and refinement, of applying a proven method to an ever wider array of microbial isolates. This framing, while powerful, also circumscribes the field of inquiry, privileging a specific class of organisms, the actinomycetes, and a specific mode of discovery, the agar diffusion assay, which inherently selects for diffusible, potent, and non-fastidious compounds. The introduction therefore presents a compelling, albeit bounded, vision for the future of antibiotic research, one built upon the solid ground of past triumphs while subtly eliding questions out fundamentally different approaches. Section 2, the methodological apparatus of discovery. The second section provides a meticulous account of the experimental procedures, presenting a refined version of the systematic screening program that became the global standard. The core of the methodology is the isolation of actinomycetes from diverse soil samples, followed by the cultivation on nutrient-rich agar, specifically a Azapic medium supplemented with yeast extract to encourage sporulation and secondary metabolite production. The central claim is that this protocol offers a reproducible and scalable pathway from raw soil to pure antibiotic-producing cultures. The evaluation of this section must focus on the rigor and the inherent biases of the techniques described. The authors detail the process of serial dilution and plating, a technique designed to yield individual colonies for subsequent testing. Each isolate is then grown as a single streak on an agar plate, and plunks of this culture are transferred to new plates seeded with test organisms, including Staphylococcus aureus, Escherichia coli, and Bacillus subtilis. The formation of a zone of inhibition, a clear area around the actinomycete plug where the test bacterium fails to grow, is the primary indicator of antibiotic production. This claim of efficacy is sound. The method is visually direct and allows for high-throughput screening of thousands of isolates. Its internal logic is coherent and practical. The procedure's strength is its systematic simplicity. The authors further describe the next steps: a cultivation of promising isolates in liquid shake flasks to generate sufficient quantities of the active substance for preliminary chemical analysis. This progression from solid to liquid culture is presented as a standard and necessary step from moving from detection to isolation. The evidence provided is procedural. The authors are offering a blueprint, and its validity is asserted through its long history of successful application in their laboratory and others. The very precision of the description, detailing incubation times of 7 to 10 days at 28 degrees Celsius, serves as a testament to its optimized and codified nature. It portrait of a mature scientific technology, a well-oiled machine for bioprospecting. Section 3: Presentation of primary results and the emergence of a candidate. This section presents the fruits of the methodological labor, detailing the screening of several thousand new actinomycete isolates. The central claim is the identification of a specific strain, designated Actinomyces geomysticus, which produces a substance with a uniquely promising antimicrobial spectrum. The authors present data in tabular form, summarizing the number of isolates screened, the percentage exhibiting any antimicrobial activity, and the smaller fraction showing activity against both gram-positive and gram-negative bacteria. The evidence consists of measurements of the diameters of inhibition zones against a panel of test organisms. The argument for the novelty of Actinomyces geomysticus rests on two pillars: its unique pattern of activity and its distinct morphological characteristics upon microscopic examination. The evaluation of this claim hinges on the completeness of the data. The paper provides clear, quantitative evidence of inhibitory action. For instance, the crude supernatant from a liquid culture of this new strain is reported to produce a 25 mm zone of inhibition against Bacillus subtilis and a 12 mm zone against Escherichia coli at a 1:1 dilution. This quantitative element lends significant weight to the claim of potent activity. The argument for novelty is supported by photomicrographs of the organism's spiral-shaped spore chains, a feature the authors contend distinguishes it from other known streptomycin or neomycin producers. The intellectual step here is one of classification and differentiation, a process fundamental to microbiology. The authors make a persuasive case that this organism, and by extension its product, represents a new discovery. The presentation is a model of scientific narration, leading the reader from a broad survey of thousands of mundane possibilities to the focused excitement of a single, promising candidate. The section effectively marshals the evidence to build a case for significance, isolating one strain from the crowd and setting the stage for its detailed biochemical interrogation. Section 4: The isolation and characterization of geomycin. Here, the authors detail the chemical procedures used to isolate and purify the active compound from the culture broth of Actinomyces geomysticus, which they name geomycin. The chief claim is that geomycin is a distinct chemical entity, a basic, water-soluble polypeptide antibiotic. The evaluation rests on the chemical evidence provided. The authors describe a multi-step purification protocol involving pH adjustment, adsorption onto activated carbon, and subsequent elution with acidic methanol. This is followed by further purification using column chromatography with an alumina stationary phase. The evidence for the compound's purity is presented through a single sharp peak on a paper chromatogram, a standard technique of the era. This evidence, while persuasive for its time, would be considered preliminary by modern standards, which would demand more rigorous methods like high-performance liquid chromatography. The argument for its polypeptide nature is based on the results of acid hydrolysis, which reportedly yielded several distinct spots on a two-dimensional paper chromatogram, corresponding to known amino acids. This is a strong piece of compositional evidence. The authors further characterize geomycin as a basic substance due to its positive charge in an electric field and its behavior during ion exchange chromatography. They provide a specific activity value for the purified compound, stating that 1 microgram of geomycin per milliliter is sufficient to inhibit the growth of Staphylococcus aureus. This quantification of potency is a crucial claim, establishing its potential therapeutic relevance. The section represents a classic piece of natural product chemistry, a demonstration of the craft of separating a single active molecule from the complex chemical soup of a microbial fermentation. The logic is sequential and deductive, moving from crude extract to a purified, partially characterized substance, with each step providing evidence for the compound's identity and purity. Section 5 Biological Activity Spectrum and in vivo evaluation. This section broadens the investigation of geomycin's biological effects, aiming to establish its potential as a therapeutic agent. The central claim is that geomycin possesses a broad spectrum of activity, with promising in vivo efficacy and acceptable toxicity in preliminary animal models. The evidence is presented in a series of detailed tables. The in vitro antimicrobial spectrum is expanded to include dozens of pathogenic bacteria, including clinical isolates of antibiotic-resistant staphylococci, as well as several mycobacterial species. The data, presented as minimal inhibitory concentrations in micrograms per milliliter, supports the claim of a broad spectrum. A particularly salient point made by the authors is geomycin's activity against strains of staphylococci that show resistance to penicillin and even streptomycin, a finding of considerable clinical importance in 1965. The argument then moves from the petri dish to a living system. The authors report results from mouse protection studies, where mice are given a lethal injection of a pathogen and then treated with geomycin. The claim of in vivo efficacy is supported by data showing a high survival rate, for example, 90% survival in mice infected with Diplococcus pneumoniae and treated with a dose of 10 mg per kilogram of body weight. The evaluation of this section requires scrutinizing the bridge the authors build between laboratory observation and potential clinical utility. The evidence for in vivo activity is compelling. The paper also addresses the critical issue of toxicity, reporting an acute lethal dose for 50% of the test population, or LD50, in mice. The value they provide, around 200 mg per kilogram via intravenous injection, suggests a therapeutic window where the effective dose is significantly lower than the toxic dose. The section is a carefully constructed argument for the compound's promise, moving logically from its spectrum of action to its performance in a whole animal system, and finally to an initial assessment of its safety profile. It represents the crucial transition in the drug discovery process from a chemical curiosity to a potential medicine. Section 6 The proposed mode of action, in a more speculative but intellectually ambitious section. Bugie and Waksman propose a mechanism for geomycin's antibacterial action. The central claim here is that geomycin functions by disrupting the integrity of the bacterial cell membrane, leading to leakage of essential intracellular components. This represents a different mode of action from established antibiotics like penicillin, which affects cell wall synthesis, or streptomycin, which targets protein synthesis at the ribosome. The evidence for this claim is drawn from a series of elegant, if indirect, experiments. The authors report that treating a suspension of Bacillus subtilis with geomycin leads to a rapid decrease in the optical density of the culture, an observation consistent with cell lyses. More pointedly, they demonstrate that this treatment causes the release of materials from the bacterial cells that absorb ultraviolet light at a wavelength of 260 nanometers, a characteristic signature of nucleic acids and nucleotides. They also report an efflux of potassium ions from treated cells. Taken together, these pieces of evidence build a coherent, albeit circumstantial, case for membrane disruption. The evaluation of this claim must recognize its inferential nature. The authors are assembling clues to deduce a mechanism. They correctly interpret the leakage of ultraviolet absorbing material and essential ions as a sign of catastrophic membrane failure, the argument is strengthened by their control experiments, which show that bacteriostatic agents like chloramphenicol do not induce similar leakage. This section demonstrates a shift from the discovery-oriented work of the preceding sections to more fundamental biochemical inquiry. It showcases the author's desire to understand their discovery at a molecular level, while lacking the definitive proof that would come from later techniques. The hypothesis is well-reasoned and grounded in sound experimental observation, representing a significant intellectual contribution of the paper. It suggests that the Waksmanian discovery pipeline could yield not only new drugs but also new tools for understanding fundamental bacterial physiology. Conclusion: the 1965 paper Isolation of New Antibiotic Substances by Elizabeth Bugie and Selman Waksman stands as a powerful summation of a mature scientific paradigm. A section-by-section evaluation of its internal claims reveals a work of exceptional methodological rigor and logical coherence, from its initial framing of the continued necessity of bioprospecting to its meticulous description of the isolation and characterization of the novel agent geomycin. The paper unfolds as a masterclass in the natural product discovery process. The claims regarding the systematic efficacy of the screening method, the novelty of the actinomyces geomycicus isolate, and the distinct chemical and biological profile of its antibiotic product are substantiated by a wealth of empirical data that was robust for its time. The authors successfully build a compelling case, moving from soil sample to a purified compound, with demonstrated in vivo efficacy and a well-reasoned hypothesis about its mode of action. The work's true significance, viewed through an evaluative lens, is its embodiment of a specific moment of scientific confidence and transition. It is the product of a research program that had transformed medicine, presented here in its most refined and codified form, yet, in its very focus on a single, successful pathway, it illuminates the intellectual landscape of its era, a landscape on the cusp of confronting deeper complexities of microbial genetics, resistance mechanisms, and the economic challenges of drug development. The paper is therefore a testament to the immense power of its author's approach and a valuable historical artifact that captures the spirit and substance of the golden age of antibiotic discovery at the very point it began to anticipate its future challenges.
We really appreciate you tuning in to this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.